Okay, so we had discussed that microorganisms have different nutrient requirements and different environmental requirements. And when they exist in the environment, they exist within community. They exist within biofilms. And then when we want to utilize them or understand them or interact with them in the medical setting, we need to be able to actually take whatever that environmental sample is or clinical sample, and then we need to be able to isolate out individual groups of organisms so that we can then learn about them, study them, identify maybe if they're the cause of disease, so on and so forth. Okay. And so the first step is often culturing those microorganisms, which you guys have done in the lab where you cultured an environmental sample, and then transferring individual colonies or individual kind of clones of organisms to new plates that you can basically isolate out, and you can see those individual colonies, and then you can do a series of tests on those individual species to see who they are or what they're doing in their environment. And so here you can see individual colonies growing on what's likely a TSA plate, which is just a general growth media. And colonies will have different morphologies, which you interacted with in lab as well. The shape of the colony, on top of the shape, also the shape of the margins of the colony. How the elevation of that colony is will kind of be dictated based on how those organisms grow within community, the size of the colony, and then texture and 3D appearance of that colony, as well as color and transparency. And so that is kind of an initial step in identifying microorganisms, but you need to continue then to utilize different methods to understand exactly what's going on with each one or be able to identify them. And so essentially, this lecture is a summary of some of the techniques that we're going to be using in our actual lab to go from pure colony, which is where you're at now, and then identify various characteristics of those colonies. Okay. And so this just gives you an idea of where samples might be collected from that are mixed, and then you want to obtain a pure culture. Now we've done this technique in lab, and I'll actually show you a resulting pure colony, which you have seen before. Here it is. That's actually from our lab. So we basically streak out concentrated streaks and then sterilize, dilute those streaks, and then you get pure cultures where you can see individual colonies and colony morphology off of that. Now in the description, you have both the resulting video where you see the plate, but also there's a video of the technique to remind you of how streak isolation actually works, where you streak an initial concentrated streak from another source, and then you sterilize your loop, and then you dilute out through subsequent streaks. So pause and watch that technique video if you want to be reminded of how we did those streak isolations. Okay. Another way to get pure colonies is what's called serial dilution, okay? And we will also be doing serial dilutions as a way of counting or numerating microbial microorganisms within a sample. And essentially, it's just what it sounds like. You take a liquid culture of microorganisms, and then you don't know necessarily, maybe you don't know necessarily how many microorganisms or what types of microorganisms. So you will actually dilute out that liquid culture into many dilutes or a dilution series, and you will actually plate through what's called the hockey stick method, because it literally looks like you're using a squeegee or a hockey stick to spread that liquid. And then you will only look at the plates that are most dilute enough that you can actually count the microorganisms or you can see individual colonies. We haven't done this method in lab yet. So what I want you to do is pause the video here, go into the description below and watch the serial dilution bacterial numeration technique video, okay? And then come back and we'll continue our discussion. Okay, so what you saw in that video is as she made the serial dilution, she wasn't sure which of those dilutions was going to actually generate a plate that had individual colonies. So her first couple of plates were what we call bacterial lawns. They were absolutely covered in bacteria, whereas the most dilute plates actually had individual colonies on them. Although sometimes you'll have a dilution which is so dilute that it doesn't grow anything. So you're always going to be choosing a balance wherever the dilution is that actually gives you individual colonies. And then you can actually see, is this a mixed culture? Is it a pure culture? And then you can continue to work with it. So we will utilize this method in lab in upcoming labs. So once we have these isolated colonies, then we oftentimes want to maintain them on various types of tests or plates in the lab, okay? And what you have, whenever you're growing a microorganism, you grow it on some sort of liquid or solid that has the nutrients that it needs to grow. And if it's a solid, it also has an agar in it that actually solidifies that nutrient base, and that allows you to see, visually the colony structures or the colony morphology of that, that growth. Or in the case of a slant, which we'll talk about in a minute, just see that there is growth. Okay. And so actually, I'm going to move forward a little bit and we'll come back to the same concept. But we have a number of different types of media that we use in the lab. We start from defined media, which is really specific, to complex media, which can be more general, even though it kind of sounds counterintuitive. And then different types of media that actually tell us different things about the metabolism of these organisms. Okay, so let's start with defined media. Defined media is literally what it says. It's like, you're working with a specific organism, or you want to grow only one specific organism from a colony, and so you put exactly the nutrients that that organism needs within a media. Okay, and so for example, here you see the exact recipe for a defined media for culturing E. coli. Now, this is not the most common type of media that we use in the lab. The most common type of media that we use in the lab is what we call complex media, media that has nutrients in it that are needed for growth of microorganisms, but they come from complex sources like soy or beef, where there's a mix of different compounds that aren't artificially mixed by a person or a human being or a recipe. They're already within that beef broth or that soy broth. There are a number of different macromolecules that organisms might utilize, right? But they're not measured out to the T exactly in an artificial method. And so this is the most common way that we will start cultures. And this is actually what we did in our lab. We started with Trypticase soy agar, which is a soy base, and it has many different nutrients that allow for general growth of microorganisms. Any media that you use, or not almost any, I won't say any media, but almost any media that you use, you could use it in different forms, a liquid form or a solid form, and you could use it in different shapes as well. So all of these are Trypticase soy agar, but one is a plate so that you can actually plate out and see colony morphology. This is a slant right here. Slants are used for long-term storage of microorganisms because plates will dry out, but slants have lids, and so they won't dry out as easily. And you literally just pour the liquid, the hot liquid agar in there, and then you just slant them, and when they dry, they solidify as a slant. You could culture microorganisms in a broth if you just want to kind of grow that colony and kind of exponential growth and get a lot of organisms, or you want to maintain it in a broth culture. That's another way. And then there's something called an agar deep. An agar deep is really useful for allowing us to see if microorganisms are anaerobic or aerobic. And so the discussion that we had in the first part of this lecture how is a microorganism kind of utilizing oxygen in its environment? You would actually just inoculate deep down into the deep with a needle all the way to the bottom, and wherever the growth is indicates to you is it aerobic or anaerobic. There's other methods as well for this, which we'll talk about. And in the description, there's a video of the inoculation of a TSA deep as well. Okay. So I would recommend that you watch that video. You're welcome to pause now or wait. Then another type, so that's general. That complex media is very commonly used, and it's just a general way of growing microorganisms. Now remember, not all, and actually many microorganisms will not grow in the lab on general media, but at least these complex media are a start. The next level is then to start selecting for specific organisms. So we'll often put inside of the media, inside of that growth, either agar or broth, something that will select for certain organisms and against certain other organisms. And that way we can actually start to generate cultures of, organisms that we're kind of looking to study, or organisms that we're looking for in our environment. So one really simple way of doing this is just to put a pH buffer in the media, and that will buffer out organisms that grow in a neutral environment versus organisms that can grow in a more acidic environment. Oftentimes, if folks are looking at fungal cultures, they'll actually utilize this method. Many fungal organisms will grow better in a more acidic environment. And this allows you to see extremophiles that are growing in very acidic or very basic environments as well. And we'll select out organisms that are not able to grow on that pH. Then we have other things that we might be looking for example, in a clinical setting. With a blood agar, this agar literally has blood cells in it. And there are certain types of pathogens that will actually lyse completely or partially blood cells. And so blood agar allows us to grow many different organisms and see, do they interact with the blood cells? No. This one right here, gamma hemolysis, doesn't. That organism doesn't interact with blood cells. It doesn't lyse them. It doesn't utilize them. Maybe like E. coli, for example. Whereas organisms that do completely lyse those blood cells, we call this beta hemolysis. Certain species of Streptococcus, they will actually completely lyse blood cells. And so you can see their presence on a blood agar plate. And then there are certain other species of Streptococcus, for example, that partially lyse blood cells. And so instead of a full clearing in the plate, which was originally just this red color, and then once the organisms grew, it was cleared out, you just get a partial clearing. And we call this alpha hemolysis. Hemolysis, hemo referring to the heme that's in blood, and lysis referring to exploding or breaking apart. Other types of selective media might select for the growth of specific organisms. So here, the phenylethyl alcohol plates, they're selective against gram-negative organisms. They'll actually kill out any gram-negative organisms. They won't grow. So you see here, Serratia marcescens, just it doesn't grow at all. There's no growth. But here you can actually see that Staphylococcus aureus is growing because that's a gram-positive organism. It's able to grow on these PEA plates, whereas gram-negative organisms like Serratia marcescens are not. Okay. Then you can add another layer to these media. You can have media that select for certain things, but then you can also have media you can add like pH indicators or different types of metabolites within the media that will tell you different things about different organisms that do grow on them, right? Kind of like the blood agar, which is telling you the difference between different organisms, but even more so, okay? And so, for example, one of the things that we'll use is the phenol red carbohydrate tests. And this is literally for each organism, you'll set up a series of like 5 or 6 tests. And each of these tubes that you see here has a different sugar in it as its kind of base metabolite. And then when you culture your microorganism inside of it, if that organism metabolizes that sugar, you will actually see through if there's acid produced because of the metabolization of that sugar or fermentation of that sugar, then you'll see a color change to a yellow because there's a pH indicator. If it's a type of fermentation that also produces a gas like hydrogen or carbon dioxide, then that will get stuck in this little tiny tube that's here inverted, and that's called a Durham tube. And so you'll be able to see that, yes, an acid is produced. You won't know which acid it is. So you just get a yellow color change for any acid, lactic acid, butyric acid. It doesn't matter, acetic acid, but you will know that an acid was produced. And 2, whether or not a gas is produced as part of that fermentation. And so if there's gas in this little Durham tube, then yes, that's a positive for gas production. If not, then it's a negative for gas production. So here, this is a test that allows us to see, okay, what sugars are these various organisms metabolizing? Then we have different types of nutrient tests to see how organisms are utilizing various nutrients in their environment. So you have some organisms that are able to break down complex structures like gelatin and starch. Here you have a plate that in the plate there's gelatin, which can be a nutrient source for organisms, and they can utilize it actually by releasing into their environment enzymes that will break down gelatin. And so here you have a positive result. You have an organism that was able to actually release enzymes into its environment and clear out the gelatin, so you kind of get this halo around it. And this one, it's able to grow, but it's not actually utilizing the gelatin, right? And so in this way, it's differential because I can tell the difference in the capacity to hydrolyze gelatin between these 2 different organisms. Okay, we also do this for starch. And so if you remember, iodine can be used as a starch indicator. If starch is present in a solution, the iodine will turn a dark black or purple color. If starch is not present in a solution, it will remain kind of a yellow amber color. And so what we'll do with the starch agar plates is we'll just grow the different microorganisms on them. And then once they've grown, we'll actually flood the plate with iodine. And if the microorganism is able to break down the starch, then in that area where the microorganism was utilizing the starch, there will be no reaction with iodine and it won't turn purple. And so we'll get a positive result that yes, this microorganism indeed does metabolize starch, and it does that via an enzyme called amylase, okay? Whereas this organism, yeah, sure, it grows, but it's not actually metabolizing the starch or producing the amylase in the way that this organism is. And so in that way, it's differential because we're differentiating between organisms that can metabolize starch via amylase and that can't. Okay, then we also have thioglycolate media. This is related to whether organisms are or not able to interact with or utilize oxygen. And so this is a broth media that has an oxygen indicator in it that will turn red in the presence of oxygen. And so you'll see this red film at the top on the tubes. It's a little bit harder to see on the tubes themselves, so I'm using a diagram here. And if you see growth just at the top where the red indicator is, then that's an aerobe. And if you see growth just at the bottom where there's no red indicator, then that's an anaerobe. And if you see some variation of that, then you're looking at variation of aerotolerance, where you might have an anaerobe that's tolerant to oxygen, or you might have an aerobe that's tolerant to anoxic conditions, so on and so forth. And so you can see where an organism lies based on their results in thioglycolate media. Okay. Continuing, you have some, so we talked about, let's pause for a second. We talked about we talked about media that is specific. It's very specific. We talked about media that's complex, so it's kind of utilized for growing different things. We talked about media that is selective, and we talked about media that's differential. But you can also have media that's selective and differential, where it selects out certain microorganisms and it doesn't allow them to grow, it inhibits their growth. And then it tells you different things about the organisms that are able to grow, which we've seen a little bit of when we looked at the blood agar. And this is another example of the MacConkey agar, where you're looking at media that is a selective only for gram-negative bacteria. So if it grows on here, it's gram-negative. Gram-positives will not be able to grow on here. And then it differentiates between the colonies. So if that has this pink halo, then that tells us that it ferments lactose. And if it doesn't, then it doesn't ferment lactose. So if it remains red or it gets kind of a yellow halo, that does not ferment lactose. And so we're able to tell the difference. Similarly, we have the mannitol salt agar, which it inhibits the growth of any organism that is not salt tolerant. And this is specifically to see if a patient has a Staphylococcus aureus infection. And so essentially, with all the salt, you don't have E. coli, you don't have the growth of other organisms that are not salt tolerant, but different Staphylococcus species will grow because the various species are salt tolerant. But if Staphylococcus aureus is present, then it'll actually utilize a sugar that's in the media called mannitol, and it'll actually ferment it. And then that fermentation will change the pH and produce a yellow color change. So essentially, what you need to know each time you're utilizing some sort of media in the lab, you need to know what is the purpose of that media, what does a positive result look like, what does a negative result look like, or what are the variations of results, and then you can then interpret that media and see what it is that you're looking at. So here we're looking at, yes, a positive for Staphylococcus aureus. This is the positive for other Staphylococcus species, but not Staphylococcus aureus, and no growth indicates that that organism is not salt tolerant. Now, generally speaking, we incubate these plates and these tubes in the same way that we have incubated them in the lab in an aerobic environment under 37 degrees Celsius. But you can actually change the temperature of your incubation if you're trying to actually see if an organism grows under lower temperature conditions or higher temperature conditions. You can also incubate these plate under various oxygen conditions. So if you the organisms that you're trying to grow are aerobic, you just incubate them as is. If they're anaerobic, then you actually need to find a way to deplete that environment of oxygen so those organisms can grow. So there's these really complex jars that you can get with with packets that will actually react with all of the oxygen in the air, and then you can grow anaerobic organisms. Or if you pause the video and go into the links with the techniques, you can see the candle jar technique, where you essentially just put your various cultures inside of a jar, and then you utilize a candle to burn out the oxygen in that jar, and then you incubate them. Okay. And that allows you to grow organisms with various aero tolerance or oxygen requirements. Okay. There's another type of media that is called a transport media, and I'll mention it here. This is essentially a media that has enough nutrients for a sample to maintain viability when it's being transported in a medical setting, right? Because sometimes you collect the sample and then it takes 24 hours or 48 hours before it gets to the lab, and then those processes, these other types of tests that we talked about, can actually be started. And so you need all of those different organisms within that sample to remain viable, so the pathogen, whichever one it is still present once the tests are actually started. And so that's what transport media is. Okay. And so with these different techniques that we've discussed now, that are your very specific, your complex, your selective, and your differential, and sometimes combinations of those. Also using transport media to get that sample where it needs to go so it can go through those various different tests. Oftentimes then you still might need to save a sample, right? Maybe you're working on different tests over many weeks in the lab, or maybe you've isolated a new organism and you need to be able to maintain it. There are various types of freezing and desiccation methods that allow you to either freeze or freeze dry microorganisms so that you can continue to work with them over months or years, okay? And those are detailed here. Okay, so that is our discussion of techniques that we use based on the information that we have about how organisms utilize nutrients within their environment and the environmental conditions, right? We play with the pH, we play with the temperature, we play with the oxygen levels, and then we're able to grow various species that we're either trying to culture, like in a medical setting, we're trying to see if there's a Staphylococcus aureus infection, or we're trying to identify. So we have a microorganism that we don't know what it is, and we want to be able to identify it. And so those various techniques allow us to do that, okay? So that content that we just covered could allow you either to do kind of your traditional notes or maybe generate a diagram or a flow chart where you have the different types of media, what it tells you, what is a positive, what is a negative. That's going to be useful for you to study both chapter 6, but also to study for our lab exam, our various lab exams in the term. Okay. So in the next discussion, what we're going to talk about is as you're culturing these microorganisms, they're growing and reproducing, and that's what you want because you want to be able to see those colonies via them growing through oftentimes binary fission, and you want to be able to see and maintain those colonies. So what's going on when you're growing these microorganisms on the various media? So first, let's review binary fission, which we've talked about, but let's just review it here.
Bacterial cells do not divide by meiosis and mitosis. Instead, most bacteria grow asexually by a process known as binary fission. In this process, the bacterium copies its chromosome, elongates, and splits off into two daughter bacteria with a genetic makeup identical to the parental bacterium. Each daughter cell can continue the doubling process. This process happens rapidly, so the number of bacteria present in a solution can increase very quickly.
Okay, so what we saw there is that it's not like, for example, in sexual reproduction in animals where the parents then have certain offspring over time and then the population kind of grows slowly. No, each individual bacterium is then going through binary fission at each round of reproduction. And so if we look at this diagram here, it's pretty common that binary fission takes about 20 to 30 minutes. So at each round, you have every bacterium reproducing in those 30 minutes, 60 minutes, 90 minutes. And so what we see is that the population will actually increase exponentially. So in the first couple hours, you'll go from 1 to about 64 cells. And then just in about 7 hours, you'll have millions of bacteria. And so that colony that's growing, in the lab, when we do our streak isolations and then we put them in the incubator for 24 to 48 hours, those colonies are trillions of cells that are accumulating in there that are then you're looking at when you're looking at the colony morphology. And so that's what you're seeing here with the logarithmic growth of bacterial colonies. Okay, and so there tend to be 4 phases that microorganisms go through, and you can see this in the macro world as well as they're growing. When you first inoculate them onto whatever media it is, there's a lag phase where they're still kind of getting familiar with that new environment. And then once they have been able to access all of the different nutrients they need within their environment, then they start to reproduce exponentially, and that's called the log phase. And then at one point, essentially, there's a balance between the number of organisms and the amount of nutrients that are present within that environment. And there's a stationary phase where the population doesn't decrease, but it kind of plateaus. And then once the amount of extraction of those nutrients is more than what is possible for that number of organisms, then the organisms tend to decline, right? And so when you're in a laboratory setting, being aware of this is important. So making sure that the conditions are correct so that a bacterial growth can actually move out of lag phase and then start to reproduce. And then if you want a long-term kind of sustenance of that colony, then you actually have to add nutrients in at the stationary phase so that you don't move into the death phase. Oftentimes what we'll do is just re-inoculate onto another plate, but there are methods like these chemostats where you have a broth inside of a kind of a large beaker or jar, and then there's actually automatic kind of addition of nutrients based on a schedule that's correct for that specific organism. Okay. Now, the next section of your book goes through bacterial enumeration and estimation. For us in our class, we're going to use a serial dilution, which we already talked about for bacterial enumeration. So you can read over this section of the text, but it's not going to be our focus. And so that actually ends for us chapter 6, where we had a discussion on microbial nutrition and growth. We talked about the requirements for microorganisms to grow in terms of nutrients and environment. That was our first discussion. Our second discussion were various types of methods that we use in the lab to grow and kind of determine what's going on in terms of metabolism with various microorganisms. And then the final discussion that we had was how organisms are growing within a lab setting and the phases that they go through as they proceed through growth and reproduction. And then being cognizant of that as you're utilizing microorganisms or microbial colonies in the lab. So that is chapter 6. I will see you in the next video. Please feel free to share with me your various flow charts and diagrams by email, and I can give you feedback about those, and that will essentially be the outlines for your studying for this chapter. See you in the next video.